So we've known that that tumors that have deficiency in the genes that are responsible for homologous recombination, the most dominant of which is BRCA1 and 2, can be measured not only by their mutational signature, but also what we call loss of heterozygosity across the genome. And so when we look at tumors that do not have BRCA1 2 mutation or other gene mutations that are part of the homologous recombination pathway, we find that there are tumors that have the same kind of signature. And so we tested this in studies that were looking at the role of PARP inhibitors in patients that were BRCA negative or wild type, but had HRD signature positive. We found that while it wasn't as good as patients that had a BRCA mutation in terms of response, we saw that it was better than those that were HR proficient. And so I always like to use the example of HRD as being a reflection that the machinery that normally governs proper homologous recombination repair is broken, and it can be represented by smoke, but we can't see the fire. We don't know where the fire is, but we know there's smoke there. So when we find and we treat the tumors that are identified by this signature, the smoke, we see a response that seems to be better than trying to treat these tumors with PARP inhibitors where there is no smoke. The best would be to see the fire, but if we can't see it, we can actually go after the smoke and try to treat those patients. So HRD is a reflection of broken machinery in the homologous recombination pathway, most strongly represented by the genes that are and the proteins that govern that at high fidelity, like BRCA1 and 2. But there are other genes and other processes that can be altered in the microenvironment, and the evidence of which we can actually see across this genome-wide loss of heterozygosity.